Well, cat breeders are aware that kittens expressing the X-linked calico coat pattern and tortoiseshell pattern are almost invariably females. Why are they certain of this? Because they breed lots of cats and they're always females. So they don't even need to know much about genetics if they're breeding cats, right? They almost never see a male tortoiseshell or calico cat. So, but a geneticist can be certain of it, right? So we should be certain of it in our class, and we don't even need to be cat breeders. And so let's take a look at why females are almost always, cats with calico cats and tortoiseshell cats are almost always females. So in class we talked about, and this is problem 7.22. So in class we talked about how, cat coat patterns are really interesting, and for the most part we only need to know about one gene. We're going to call this gene C, and this gene C has two major alleles, one which confers the red pigment, which is kind of that orange color. I don't know, breeders call it red. Kind of looks kind of orangish to me. So a red color, and allele CB, which is for a black color. So a red pigment or a black pigment. And then you have gray and things like that, and tan, and those are all due to other genes which sort of dilute these pigments and lead to different shades of black or red. Okay, so but we're only concerned with the red pigment and the black pigment. Okay, so these happen to be X-linked. This is why we're talking about it in this chapter. So these are X-linked. And as a result, if you have, let's say our male genotypes can be CR with a Y chromosome, CB with a Y chromosome, and that's it. And these males here are always going to be sort of that red-orange color, or maybe they have the orange base. But okay, so red or orange, and these are always going to have black. These are always going to be black, so they're never going to be orange and black, like with a calico or tortoiseshell. So females, however, can be CR, CB, CB, or CR and CB. Now these are always going to be red. There's no choice there, right? There's no black allele. These are all going to be all black. So all red up here, all black down here. And these are the ones we're interested in. So notice how these are heterozygous, and males can't be heterozygous for X-linked genes. They are hemizygous. So these females right here have both alleles, one to produce red, one to produce black. And it's hard for us to know which is dominant over the other one because of this thing called X inactivation that we just talked about in some of the earlier problems. So X in inactivation. So in each cell, one of these is going to be inactivated. So during the first 1 to 3 weeks, and maybe that time might be a little different for cats, neither X is going to be inactivated, but then we're going to end up with lots of cells, thousands of cells, with both of those activated, and then one of these chromosomes, X chromosomes, is going to be inactivated. And let's say in this one and this one, the X chromosome with the black allele is inactivated, and it's put into the bar body. And the red allele is still active because it's on the active X. And in these down here, we'll say the X chromosome carrying the red allele is inactivated. And the one carrying the B allele is still active. Okay, so every time these cells divide from now on, the black allele, this one right here, the X chromosome with the black allele will always be inactivated. Every time these cells, every time a cell with the black chromosome inactivated, with the X chromosome carrying the black allele is inactivated, the daughter cells will also have the black chromosome, the X chromosome carrying the black allele inactivated. Okay, so same thing for cells which have the X chromosome carrying the red allele inactivated. So I've diagrammed the nuclei here. So let's say that undergoes mitosis, and we've now got 2 cells with nuclei here, and each time the X chromosome with the red is inactivated. So once an X chromosome is inactivated, it stays inactive in all of the daughter cells following each mitosis. So if you have a cat here, here's our cat, and if you've been coming to class, you know I'm getting a little bit better at drawing these little animals. Here's a tail. Okay, here's our cat, and if we have a patch here with a bunch of cells, all these cells are probably, the closer the cells are together, the more likely they came from the same parent cell. So that's why we can have a patch of cells here, pigment cells, that all produce, let's say, black. So we got a black patch there. Now let's say there's a patch over here. We have a bunch of cells that all started from a cell that had the red allele, no, the black allele inactivated. So all these would be red. So, and we could do the whole cat, we could do the whole thing to the whole cat that way. And this would be a calico cat where the patches are nice and big, and the reason the patches are nice and big is there's another gene which kind of slows the rate of migration of the cells and keeps them from far apart from one another. So this is why we don't have small patches of red and black in the calico. But in the tortoiseshell, well, they don't have that particular, that other gene that controls that slows the rate of migration. So you can have little patches of black next to little patches of red next to little patches of black, and that's a tortoiseshell cat. And you should look up some pictures of tortoiseshell versus calico to see what I'm talking about. But the colors, the patches are closer together in the tortoiseshell. But the concept is the same. We have X chromosome inactivation leading to the patches of red and black. And why can't we see tortoiseshell or calico in a male? Well, it's because males only have one X chromosome. So that same X chromosome is going to be active in all cells of the male. So let's go back up to the male genotypes. No X chromosomes are inactivated in the males, so they can only be all red or orange or all black. Now there is the rare case of the Klinefelter cats, right? Klinefelter, which are X, X, Y. Now in this case, you could have an X chromosome that has the CR allele, and another one that has the CB allele. And these cats right here would be either tortoiseshell or calico. However, they would be sterile because Klinefelter, the syndrome, is associated with sterility too. So you can occasionally get a Klinefelter cat, and so once in a while you will see a male calico or tortoiseshell. And I sent a story out about how a vet, I think it was 54-year career, just recently saw his second male calico. So it's really interesting. Okay, so this is that's the answer to 7.22. Okay, so I haven't rehearsed this yet, but let's see if we can do it with one shot here. So we're going to have 8 RNA polymerases in our transcription factory. So those little dots that I was diagramming up there, let's zoom in on one of these. Because it's one of those dots. Okay, this is our transcription factory, and we're going to draw 8 different RNA polymerases. Let's hope this is right. We're just going to put them as circles here. 1, 2, 3, 4, 5, 6, 7, 8. Okay, so we're going to have a chromatin loop come out here. Now this is coming from a chromosome territory. And I am going to draw it going through this polymerase right here. Okay, so let's say the promoter region, this is a double-stranded DNA, right? Okay, promoter region right here, let's say the +1 is this way, and let's say the RNA polymerase is moving this way. We have a little transcription bubble here. The strands are separated, and we have, let's see, transcript coming out here. Okay, that looks pretty good. What did we leave out though? I think we left out like mediator and the transcription factors and things like that here. We left all that stuff out, but I think that's a pretty good start for one of these genes. Okay, let's try another one up here. Let's see if we can put mediator in here. So I'm going to put a loop like this. Okay, so a big loop here, and okay, there's the RNA polymerase. And we could put lots of things in here, right? So I'm going to put nucleosomes. Again, the diagram should probably be much more complicated than this, right? Nothing's really to scale. We're not showing the DNA wrapped around the nucleosomes. But okay, let's have our enhancer right here. Now the problem didn't ask for enhancers, but I'm going to put one in anyways because I think it's kind of fun. So, and we'll have transcription, specific transcription factors bound here. We'll have the mediator complex bound here. Name that M. And we'll say, let's say this is the promoter region right here, and say this hasn't started transcribing yet. Promoter. Okay, we'll have the general transcription factors here. And again, we don't have to memorize these. There's too many of them, and we'll worry about what things we need to memorize in molecular biology 350 if you're going to take that. So we just, in general, want to know how these things work for this 200 level genetics class. So general transcription factors, and they're interacting with mediator here, and mediators interacting with the specific transcription factors, and those are bound to the enhancer. And so we got this nice chromatin loop formed, and we're loading the RNA polymerase onto the loop, and the RNA polymerase is going to, let's say, transcribe that way. Okay, or maybe the DNA is going to come out this way, and the RNA polymerase will stand still. Who knows? But okay, so this is another transcript is being made right here. So we could do the same sort of thing for every one of these RNA polymerases, and we have a transcription factory with different loops coming out of the chromosome territories into the transcription factory. So really cool stuff. Again, this is just a model. Scientists are still trying to figure out how all this stuff works, but very, very fascinating stuff. I hope you think so too.